Hey everyone, in this video we are going to talk about the Trip Operon, Tryptophan Operon. Now remember, in the last set of videos we talked about, or we discussed the Lac Operon. So the Lac Operon is an example of an inducible system. Now, why is it an inducible system? So the effector molecule, in this case, which is lactose, effector molecule, in this case, is lactose. Now, when it binds to the repressor, which was Lac I, when it binds to the repressor, Lac I releases the operator, right? Releases the operator. As a result, the Lac Operon, the Polycistronic Rna, the Lac Operon is transcribed. So we could also say the Lac Operon is induced, and that's why the Lac Operon is an inducible system. Now, the Trip Operon, the Tryptophan Operon, is an example of a repressible system. Repressible system. Now why? Why is this a repressible system? So in this case, the effector molecule, which is tryptophan, the amino acid tryptophan, now, when it binds to the repressor molecule, or protein, I should say, which we will soon learn is called Trip R. So when the effector molecule, tryptophan, binds to the repressor protein, Trip R binds to the operator. So the Trip Operon is repressed. Maybe a better way to say that is not transcribed. Now, so what is an effector molecule? In both of these cases, we can see the effector molecule is something that binds the repressor protein. So effector molecule is a thing, you could say a molecule, but a thing that binds to the repressor. So with a Lac Operon, it's lactose. With a Trip Operon, it's tryptophan. And how the operons react when the effector molecule binds to the repressor designates whether it's an inducible operon or a repressible operon. So the effector molecule, when it binds to the repressor, the Lac Operon is induced, it's transcribed. Therefore, the Lac Operon is inducible. The opposite occurs with the Trip Operon. When the effector molecule, tryptophan, binds to the repressor protein binds to the operator, and the Trip Operon is not transcribed. So the Trip Operon is an example of a repressible system. Now let's take a closer look at the Trip Operon. So Trip Operon, now it looks something like this. We have, well, this isn't the operon over here. This gene, similar to the Lac I repressor, this is going to be the gene for Trip R, the repressor protein. Now it's not connected to the operon, found somewhere else in the genome, and someday I'm going to look at the E. coli genome and see exactly where this is relative to the operon. But I'm going to put these little hatch marks here to indicate that it's not necessarily next door to the operon, somewhere else in the genome. And let me diagram the Trip Operon here. So we're going to call this P for the promoter region. This will be O for the operator region, and then we're going to have this L region. This would be called the leader region. We'll talk about this after, at the end of this video. The structural genes, remember, structural genes are any gene that doesn't encode a product that influences expression of another gene. We talked about that in the last set of videos. So the structural genes are Trp E, Trp D. It's easy to remember the order of these because they just go backwards from E. Trp C, Trp B, and Trp A. So essentially all of these genes encode biosynthetic enzymes needed to produce tryptophan through a biosynthetic pathway. Now, again, I'm not exactly sure where the +1 site is. I'm going to assume it's right here, let's say between the promoter and operator region. It doesn't really matter exactly where it starts. So when this is transcribed by RNA polymerase, we get a polycistronic RNA. And in the last set of videos, we learned a cistron was a region of a gene or an RNA that encodes a protein. And a polycistronic RNA is something that encodes several proteins from one RNA molecule. So let's say this is where Trp E, this Trp E cistron is here. Let's say D is here. We have start and stop here, start and stop signals there. So we can get 5 different proteins made from this polycistronic RNA. Start, stop, start, stop, start, stop. Now this would be the 5' end, 3' end, and we're going to talk more about this leader sequence in a moment, and this would be the 5' UTR, untranslated region. 3' UTR right here, 3' UTR. So that's the Trip Operon, and the repressor in this case is going to look, I'm going to diagram it a lot like how I diagrammed the Lac I repressor for the Lac Operon. So we're going to have a little binding site for tryptophan right here, and I can diagram tryptophan like this. It looks a lot like lactose, huh? So if this is Trp R, now when tryptophan binds to Trp R, it undergoes an allosteric shift to something that looks like this, at least conceptually, right? So tryptophan bound induces a change in the conformation, which opens up the operator binding site. So it's easy to see when tryptophan levels are high. Let me see. We have low tryptophan versus high tryptophan. 2 conditions. When tryptophan levels are low, the repressor will look like this, right? And it can't bind to the operator. So it won't be able to block transcription of the operon by RNA polymerase. So when tryptophan levels are low, the operon will be transcribed. We will make all of these biosynthetic genes to make tryptophan. Because when tryptophan levels are low, the cell needs more tryptophan, needs to make it. Now, if tryptophan levels are high, let's say there's lots of tryptophan in the environment, wherever E. coli finds itself, it doesn't need to transcribe this operon, make these biosynthetic genes. It doesn't need to make more of its own tryptophan. It can get it from the environment. So when tryptophan levels are high, tryptophan binds to the repressor, it opens up the operator binding site, Trp can bind to the operator and block transcription of the Trp Operon, which is great because, you know, E. coli doesn't want to transcribe this if it already has enough tryptophan. So there's another level of regulation on top of this repressor binding the operator, and it's called the Trip Attenuator. And the Trip Attenuator is found in the leader region. And I believe in the notes I had said that let's just call the leader region and the attenuator region the same thing. We'll consider them one and the same. And that's fine for this course and for the exam. There are some researchers that consider a specific region of the leader region to be the attenuator, and I'll address that in a moment. So let's take a quick look at this leader region and what it means to be an attenuator with respect to prokaryotic gene expression. So let me sketch here the operon. Here's our promoter. Let's say that's our +1 site right there. Operator. Now here's the leader region. I'm making it kind of big. You know what? I'm going to have to make it even bigger down here. So, and let's say Trip E starts here. We'll call that L for leader. Now, when RNA polymerase binds here and starts transcribing, well, I can diagram that RNA right here. So let's say RNA polymerase is transcribing this RNA right here, and I probably should have diagrammed it up here on this DNA, but let's just look at the RNA level that RNA polymerase is transcribing from the Trip Operon. So this RNA, now this leader region, I want to kind of zoom in on this right here. So say I'm zooming in, I'm zooming out actually right here, because I want to, there's a lot of parts of this leader region on this RNA that I want to talk about. So if this is the 5' UTR, now the leader region is going to start right here at this AUG. So it turns out that even before Trip E is translated, the Trip E is translated into a protein, there's a little peptide that is translated from the messenger RNA corresponding to the leader region. And the start codon is going to be right here at the beginning of the leader region. And then over here, a little further down, there are going to be 2 tryptophan codons, trip codons. Then over here in these regions down here, well, we don't really care what the nucleotides are here, but I'm just going to label them 1, 2, 3, and 4. So 4 regions called easy to remember, right? Just 1, 2, 3, and 4. And then right here at the end of the leader region, there are going to be 6 U's. So we're going to call this the poly-U region. Now, all of these parts of the leader region work together to help regulate whether RNA polymerase keeps transcribing the operon or it stops and terminates transcription after or at the poly-U region. So all of these work together. I'm going to go over how they work together to control that. And whether or not RNA polymerase stops after this U depends on the levels of tryptophan in the cell. So, and another thing I wanted to point out is some scientists consider this the attenuator, or some references. Some references will consider the whole region the attenuator, the whole leader the attenuator, and others just say this poly U region of the leader sequence to be the attenuator region or the attenuator sequence. Okay, so how do these work together to, control whether RNA polymerase continues to transcribe the operon after the poly U region? Well, okay, so in order to get to this point right here, RNA polymerase, it had to make it by the operator. It had to make it by here. But let's say it's possible that it made it by when tryptophan levels were high. So E. coli has another level of regulation to decide whether or not it stops after here. So we have to assume that, this is important because sometimes RNA polymerase can make it by the operator even when tryptophan levels are high. So E. coli has another method, has the Trip Attenuator to add another level of control to prevent RNA polymerase from transcribing the operon when it doesn't need tryptophan. Okay, so first, I think I need a sharper pencil for this. Am I still good? A couple times I have, trying to make this video, and then I look up 15 minutes into the video and see I wasn't recording. Oh no. But no, we're recording. Okay, so let's consider the condition where tryptophan levels are low. So low tryptophan in the cell. Now, here's the RNA, the nascent RNA, and maybe you remember we consider nascent RNA to be still undergoing transcription. I'm going to diagram a little hairpin loop right here. What does that mean? So that means that the ribonucleotides on this strand over here are complementary to the ribonucleotides on this strand. So the RNA can fold into that little loop structure right here. And let's say this is where RNA polymerase is. I draw it like a little Pac-Man. And let me see, I can put the DNA in here like this. And I'll have the DNA. This is the transcription bubble. It's a pretty nice bubble. The DNA just comes off going this way. Okay, now let's say this is the 5' UTR, the untranslated region. And then we have the leader sequence starting here. And let's say ribosomes start translating, so they load over this AUG of the leader. They load on there and they start translating a peptide. And then they're going to get to those tryptophan codons. So I'm going to draw those in like right there. I'm not going to actually draw the codon, the nucleotide sequences there. But I will do this and say trip codons. So there are 2 trip codons right there. Now region 1, I'm going to say is right here. So let's say this is region 1, and the ribosomes are covering, the ribosome here is covering region 1, or part of region 1. Now region 2 is this part of this hairpin loop right here. Region 3 is right here. And region 4 is going to be right here. And that UUU is going to be right here. 1, 2, 3, oh, 6 U's, right? Poly U is right here. Okay, so when tryptophan levels are low, what happens is the charged transfer RNAs, or charged tryptophan transfer RNAs, what does that mean? Remember that a charged tRNA has tryptophan attached to it. So a tRNA with the anticodon for tryptophan that has the amino acid linked to the tRNA, that's considered a charged tRNA. So when these levels are low, the ribosome stalls on these tryptophan codons. And as a result, it occupies or covers part of this number 1 region in the leader sequence. And I can start showing the little peptide that's being made here from the leader, because it started translating and then it stalled at the trip codons because charged tRNA levels were low, because tryptophan levels are low in the cell. It occupies part of the 1 region. As a result, regions number 2 and number 3 pair up. And when these are paired, 3 does not pair with 4. So we're going to see that in the next example when tryptophan levels are high. So when 2 and 3 are paired, 3 does not pair with 4, because 2 and 3, they're more attracted to each other than are 3 and 4. Now when 2 or 3 are paired, this hairpin, this is called the anti-terminator hairpin. Anti-terminator hairpin. And I kind of want to do an Arnold Schwarzenegger joke there, but I'm not going to do it. Okay, so 2 and 3, when these are paired up, this is the anti-terminator hairpin. When this is formed, RNA polymerase does not stop transcribing at this poly U. It keeps going. So it's going to now transcribe the rest of the operon. And all of those biosynthetic genes for making tryptophan will be transcribed. Now that's when tryptophan levels are low. So when tryptophan levels are high, something else happens, right? So you can probably predict what's going to happen. The ribosome's not going to stall, right? And as a result, it's going to cover 1 and 2. It's going to cover 1 and 2, freeing up 3 to form a hairpin with 4. And that's going to be called the termination hairpin. So let me diagram that. So I'm going all the way over here. Now here's our hairpin. Now here's the U. Now RNA polymerase has fallen off the template somehow because of this 6 U's right here. So I can diagram the DNA. Now the transcription bubble is gone. Transcription has been terminated. Here's the AUG. Here's the 5' UTR. Now in this case, these are the tryptophan codons. The ribosome blew right by those because there was plenty of tryptophan in the cell and plenty of charged tryptophan tRNAs. So now regions 1, well, that's not a good place to diagram that, but I'm going to put 1 right here. 1, and I have to make this a little bigger so it occupies part of 2 also. So 1 and 2 now, regions 1 and 2 are covered by the ribosome. Remember, the ribosome is a huge complex here. So it's covering regions 1 and 2, allowing 3 and 4 to form the hairpin. This is the termination or Terminator Loop. When this is formed, RNA polymerase stops transcription after this U right here. 1, 2, 3, 4, 5, 6, 6, not 7 of those guys. Okay, so we can see how, you know, we have a second level of regulation. You know, if tryptophan levels were high and somehow RNA polymerase made it by the operator region, maybe the repressor wasn't bound for some reason, made it by the operator and was able to transcribe this leader right here, well, the ribosomes would jump on, and remember, ribosomes can begin translation while RNA is being transcribed in prokaryotes because there's no nucleus. Ribosome hops on, blows right by the tryptophan codons, is able to incorporate tryptophan into this growing peptide. and it occupies regions 1 and 2, allows the Terminator hairpin loop to form, and that causes RNA polymerase to be dissociated after transcription of this poly-U region. Why? What is it about this structure that causes RNA polymerase to dissociate? I don't know. I don't know if scientists know yet. Maybe they have some ideas on why it happens, but we're not going to go over how it happens because I don't really know how it works, I'm not even sure anyone really knows how it works. So we know what causes it, but the mechanistic explanations for why it dissociates, I'm not really sure. So I hope that helps you understand how the Trip Attenuator works, and you have an overall understanding of how the Trip Operon works and why it's a repressible system and not an inducible system like the Lac Operon. And in the next videos, we are going to take a look at eukaryotic gene expression. Okay, see you in the next videos.